Welcome back to the Deep Dive. Today we are taking on a monumental topic, translating the entire unit on plant form and function.
It's a big one.
It is. We're going beneath the soil and way up above the canopy to find out how plants, which can seem so passive, are actually, dynamic masters of engineering, resource management, and defense.
It's the perfect topic for a deep dive, really, because it fundamentally changes how you view the world around you.
Right.
We're going to try and give you a shortcut to understanding all these complex biological mechanisms that drive plant structure, their growth, how they get resources, and even their really sophisticated sensory and chemical defenses.
And to kick off this massive journey, we're going to use a phenomenal story that really shows the stakes of plant biology and the power of human intervention.
Yeah, this is a great one.
We're heading to Hawaii in the 1990s, where the entire papaya industry was on the brink of collapse, and one scientist, Dr. Dennis Gonsalves, stepped up to save it.
And Dr. Gonsalves' background is amazing. He was a local kid who ended up becoming a plant pathologist. The crisis he faced was the papaya ring spot virus.
Yeah.
This virus was just devastating because it was rapidly transmitted by these tiny sap-sucking insects called aphids.
And the aphids didn't need to spend long on the plant to be vectors, did they?
Not at all. We're talking less than a minute.
Wow.
An aphid would feed, pick up the virus, jump to a healthy plant, and the cycle would just repeat exponentially. The symptoms were grim. You'd get these ring spots on the fruit, yellowing leaves, and severe stunting that often meant the plant never produced usable fruit.
So it's wiping out entire farms.
Completely.
So he tried the classical virology approach first,
Right.
using a mild strain of the virus to protect against the harsh one.
He did, but it failed to provide reliable protection out in the field. It just wasn't consistent enough. And this is the moment where the science really pivots. It is. Gonsalves realized classical breeding was way too slow, and traditional virology wasn't working fast enough to keep up with how fast this thing was spreading. So he took the plunge. He retrained himself as a molecular biologist, really embracing the new world of genetic engineering.
And the solution they developed utilized a piece of equipment that sounds straight out of a sci-fi movie. I'm talking about the gene gun.
It's essentially what it was, yeah. The goal was to give the papaya a permanent built-in resistance.
Right.
So based on research that showed introducing a viral coat protein gene into the host genome could induce immunity, they took that specific DNA, the blueprint for the virus's outer shell, and they coated tiny tungsten beads with it.
So they're literally weaponizing the virus's own code against it.
That's a great way to put it. And then they shot those tiny DNA-coated beads directly into papaya embryos that were growing in culture.
They're just blasting the genetic instructions into the plant cells.
Precisely. And hoping, that some of it would integrate into the papaya genome. Out of thousands of these experiments, they found a single line that grew up completely resistant to the ring spot virus.
Just one?
One successful line. And that single resistant line became the parent of the rainbow variety of papaya. This was developed and ready just as the virus was wiping out the main growing region of Puna.
So they literally rescued the crop just in time.
They did. The survival of the entire industry hinged on understanding plant function, structure, and the strategic application of that genetic knowledge. And that, in a nutshell, is the theme of our deep dive.
Right.
The interplay between the plant blueprint and how we manage it.
Okay, so let's unpack this blueprint.
Yeah.
We have to start with the fundamental constraint of plant life, which is that they're immobile. They're rooted between two very, very different environments.
And that one constraint defined their entire evolution. I mean, think about it. They have to acquire light and carbon dioxide from the atmosphere above.
Right, from the air.
And then water and minerals from the soil below. This split necessity led directly to the two distinct organ systems. You've got the root system below ground and the shoot system, stems and leaves, above ground.
And neither system can survive without the other, can it?
Not at all. The roots, which usually aren't photosynthetic, they need sugars imported from the shoot system.
The food factory.
Exactly. And the shoot needs the massive volume of water and minerals that are absorbed by the roots. It's an incredibly close mutual dependence.
So let's focus on the roots first. Their functions are pretty straightforward, but critical.
Oh, absolutely. Anchoring the plant, absorbing resources, and often storing carbohydrates for later. And their efficiency at absorption is all about maximizing surface area.
So how do they achieve such a huge surface area? It seems like a simple root wouldn't be enough.
It wouldn't. Primarily, they do it through these tiny extensions of the epidermal cells called root hairs.
Okay.
And if you could somehow lay out the surface of all the root hairs on a mature corn plant, You would need hundreds of square meters. It's an incredible amount. And on top of that, virtually all plants utilize what we call mycorrhizal associations. These are symbiotic fungi that extend the absorption network even further out into the soil. So the roots are truly specialized machines for finding and extracting resources.
Okay, moving above ground, the stem is, I guess, the support structure and the conduit, the highway.
Right. And the leaves are the photosynthetic engines and the site of gas exchange.
If you look closely at all these different organs, they're all built from three basic continuous tissue systems that run throughout the entire plant body, linking everything together.
The first one is the dermal tissue system. You can think of it as the plant's protective barrier.
Like its skin.
Exactly like its skin. In non-woody plants, this is the epidermis, and it's often coated with a waxy cuticle.
Which is all about preventing water loss, right?
Absolutely, a necessity for minimizing water loss in the dry atmosphere. Specialized components of this dermal system are the root hairs we just mentioned, and on the shoot you have these things called trichomes.
What's the story with trichomes? They sound fascinating.
They are. They're basically plant hairs, but they have extremely varied functions, sometimes even on the same leaf. Some are just physical defenses, little spear-like structures that can hinder crawling insects.
A physical barrier.
A physical barrier. Others are tiny secretory glands, and they release protective chemicals or oils that deter herbivores or can reflect excess UV damage. They really are the plant's first line of defense against the elements and predators.
Then you have the vascular tissue system, the plumbing.
The plumbing, yes. Xylem and phloem. Xylem is the upward flow, water and minerals from the roots. Phloem is the more complex bidirectional flow. It distributes the photosynthesis, the sugars, from where they're made or stored to where they're needed.
And these form the veins in the leaves.
The veins in the leaves, the rings in a tree trunk, that's all vascular tissue.
And the third system, the ground tissue system, is kind of the filler, right?
Exactly. It's everything else. But it's not just filler. It's specialized for storage, for support, and for photosynthesis. If you look at a cross-section of a young stem, we differentiate the ground tissue based on its location. The tissue that's internal to the vascular bundles is called the pith.
Okay, pith in the middle.
And the tissue external to the bundles is the cortex. Within this ground tissue, you have cells that specialize into, say, parenchyma for storage and metabolism, and then tougher supportive cells like collenchyma and sclerenchyma.
So we've laid out the static structure. But plants are defined by their capacity for indeterminate growth. They grow their whole lives. They don't just stop after adolescence like animals do.
This perpetual growth is driven by these special regions of unspecialized dividing cells called meristems. And this is also how we categorize the life cycle of flowering plants. You have annuals that complete their cycle in 1 year, like rice.
Right.
You have biennials, which need a 2-season cycle, like carrots. And then you have the really long-lived perennials.
And some of these can live for an incredible amount of time.
The sheer duration is mind-boggling. Some clones of buffalo grass are genetically linked to plants that have survived since the last ice age, something like 10,000 years ago.
That's amazing. So when we talk about growth, we need to distinguish between growing longer, which is primary growth, and growing wider, secondary growth. Primary comes first.
Primary growth is all about vertical extension, making roots go deeper and shoots grow taller. And it is controlled by the apical meristems, which are located at the very tips of the roots and the shoots.
Let's focus on the root tip structure for a second, because it's a beautifully organized system. It's designed to push through abrasive soil without damaging its vital cells.
It is a microscopic marvel. The very tip of the root is protected by the root cap. It's constantly sloughing off cells, and it secretes this polysaccharide slime.
Lubricant.
A lubricant, exactly. It eases the passage of the growing root. And just behind that cap, growth occurs in 3 overlapping zones.
Okay, what are those 3 zones doing?
First, you have the zone of cell division, which contains the apical meristem itself, constantly generating new cells. Then, just behind that, is the zone of elongation. This is where the major increase in length actually happens.
The cells are stretching out?
They swell up and physically push the root tip further into the soil. And finally, you have the zone of differentiation, where the cells start to specialize into the various tissue systems we just discussed, dermal, vascular, and ground.
And what triggers branching in the root system? Where do those lateral roots come from?
Well, lateral roots, which are also a form of primary growth, they don't just pop out on the surface. They actually originate deep inside the root from a layer called the pericycle.
Which is part of the vascular cylinder.
It's the outermost layer of it, yes. So they literally have to push their way through the cortex and the epidermis to emerge, which ensures they're connected directly to the central vascular tissue right from the start.
Now, in the shoot system, branching is controlled by axillary buds, and this leads to the concept of apical dominance.
Right. Apical dominance is the phenomenon where the active shoot apical meristem chemically inhibits the growth of the lateral axillary buds that are below it. This is why many trees grow in that classic single, tall spire shape.
And if you cut off the top, it gets bushier.
Exactly. If you remove the apical bud, if a deer eats it, or if you're pruning a hedge, that inhibition is lifted. The axillary buds activate, and you get a much bushier, fuller plant. It's an immediate demonstration of hormone control over structure.
Okay, so primary growth is length. Secondary growth, driven by lateral meristems, is all about girth. This is what woody plants do.
This is how trees get thick. The two lateral meristems are the vascular cambium and the cork cambium. The vascular cambium is the real production factory. It lays down secondary xylem inwards, which is what we call wood, and it lays down secondary phloem outwards.
And this secondary xylem gives us the classic growth rings. And those rings, they tell a detailed story, don't they? It's much more than just the age of the tree.
Absolutely. The difference between the early wood, which is formed in the spring and has these large cells for maximum water transport.
For the new leaves.
For the new leaves, exactly. And the late wood, which forms in summer and autumn and has smaller, thicker-walled cells for mechanical strength, that difference creates the distinct rings, and this pattern is the key to dendrochronology.
Tree ring daving.
Exactly. By analyzing the width and the density of these rings, scientists can accurately reconstruct climate history, recording past droughts, freezes, or high rainfall years, sometimes centuries into the past. It's essentially a plant-written history book.
So while the vascular cambium is building the wood structure, the cork cambium is maintaining the outer defenses.
That's right. As the trunk gets thicker, the original epidermis inevitably ruptures. So the cork cambium replaces it by producing the periderm, that tough, protective outer layer. The cork cells it produces are crucial. They deposit a waxy substance called suberin into their walls, and then they die, forming an impermeable, rot-resistant layer.
And the term bark is botanically defined as everything outside the wood.
Right. Correct. Bark is defined as all tissues external to the vascular cambium. So that includes the secondary phloem and all the layers of the periderm, which is the cork cambium and the cork cells it makes.
And just to wrap up this section, we have to touch on developmental plasticity. The source material makes a really powerful point that a plant's structure isn't fixed. It adapts based on its environment.
It's an amazing adaptation. It's the ability of a single genotype to produce dramatically different phenotypes based on external conditions.
Right.
A great example is Cobumba aquatica, the fanwort. The leaves that grow underwater are fine and feathery, which minimizes resistance to water flow. But the leaves that reach the surface are broad pads, maximizing light capture and flotation.
Same DNA, totally different forms.
Exactly the same DNA. The environment dictates their final form.
So how do the cells inside the plant know what to become? Is it based on their ancestry, their lineage, like it is in animals?
Not primarily, no. Cell differentiation in plants is driven mainly by position-based mechanisms. A cell's final fate is determined by the signals it receives from its neighbors. Where it ends up matters more than where it came from.
So location, location, location.
It really is. We see this with root hair formation. An epidermal cell that's touching two underlying cortical cells gets signaled to differentiate into a root hair cell. But an epidermal cell that's only touching one cortical cell remains hairless. And this decision is controlled by the expression of a gene called GLABRA2. It's highly precise signaling based entirely on spatial location.
All right, now that we understand the structure, let's explore how the plant's plumbing actually works, the dynamic systems of resource acquisition and movement.
And we have to start with the evolutionary trade-offs. I mean, why be tall? Well, for light competition. But being tall demands this massive investment in thicker stems secondary growth, which needs more energy.
It's all a budget calculation.
Everything is a budget calculation. And the most classic budget crisis for a plant is the photosynthesis water loss compromise.
I was just about to bring that up.
It's fundamental. You need broad leaves and you need open stomata to maximize your carbon dioxide uptake for photosynthesis. But every single time a stoma opens, water vapor rushes out.
And a lot of it.
Over 90% of the water absorbed by the roots is lost through the leaves via transpiration. The plant is in a constant balancing act between starving from low CO2 and dehydrating from high water
I
loss.
also find the adaptability of the root architecture fascinating. They aren't just mindless probes. They actively seek out nutrients.
They are intelligent in their growth patterns. If a root system hits a nutrient-poor patch of soil, it will just tunnel straight through it. But if it hits a high nitrate patch, it immediately begins extensive branching right there in that specific area to maximize the capture of that resource.
It's actively foraging.
It's foraging, and its cells respond by synthesizing more nitrate transport proteins to increase the uptake efficiency. It's incredibly dynamic.
So let's talk about movement. We have short-distance movement, cell to cell, and then the long-distance bulk flow.
Right. Short-distance movement can happen through a couple of routes. There's the apoplastic route, where substances move along the outside of cell membranes, through the cell walls and the extracellular spaces, without ever entering the cytoplasm.
The outside route.
The outside route. Then there's the symplastic route, where a substance enters one cell and then travels through the connected network of cytoplasm, which are linked by these little channels called plasmodesmata.
So it's moving through the inside of the cells.
Exactly. But for massive transport over meters in a tall tree, you need bulk flow.
Right. Diffusion is not going to cut it.
Not even close. Bulk flow is driven by pressure potential, and it moves the entire solution rapidly. It's vastly quicker than simple diffusion. And this is the domain of the xylem and the phloem.
So let's tackle water transport in the xylem, which defies gravity, using the cohesion-tension hypothesis. This is always the wow moment for plant physics for me.
It truly is. The physics are so counterintuitive. Water is not pushed up a tall tree, it is pulled. We do see a minor upward push, something called root pressure. It's created when minerals are pumped into the vascular cylinder, causing water to follow osmotically. And this sometimes leads to more inundation. You see little water droplets on leaf margins. But this force is way too weak for any tall plant.
So the main engine is the transpirational pull that originates way up in the leaves.
Yes. When water evaporates from the moist cell walls inside the leaf mesophyll, that air-water interface creates immense surface tension. It's a negative pressure potential, or suck. And this tension is transmitted downward through the entire column of water in the xylem.
And what stops that water column from just breaking under all that tension?
Water's own inherent properties, cohesion and adhesion.
Right.
Cohesion is the strong hydrogen bonding between water molecules. It ensures that when one molecule evaporates from the leaf, it pulls the next molecule up.
Like a chain.
Exactly like a chain. And adhesion, which is the attraction of water to the hydrophilic xylem cell walls, resists the pull of gravity and helps prevent air bubbles or cavitation from forming and breaking that chain. The xylem itself is perfectly structured for this. It's just dead, hollow cells, tracheids, and vessel elements that offer minimal resistance.
But before water even gets to the xylem, it has to pass a crucial security checkpoint in the root, the Casparian strip. Why is that barrier so important?
It's the gatekeeper. It's located in the endodermis. The Casparian strip is this waxy, impermeable barrier that's embedded in the cell walls, and it blocks the apoplastic route, the outside route.
Okay, so it forces a decision.
It forces a decision.
Yeah.
Any water and minerals that were moving through the cell walls are forced to pass through the plasma membrane of an endodermal cell, switching to the symplastic route. And this ensures the plant can control exactly which solutes enter the highly sensitive vascular cylinder. It's a selective filter mechanism.
So since this transpirational pull is the engine, regulating the water loss via stomatal control is absolutely essential.
The stomata, the pores, flanked by guard cells. They're the valves. They open when the guard cells become turgid. That means they swell up, causing the cells to bow out and open the pore.
And how do they swell up?
That turgor is achieved by actively accumulating potassium ions, K plus argos. It's a process fueled by proton pumps. Water then follows the K plus osmotically, and bang, the stoma opens.
So the plant has a molecular pump driving the physical opening of the valve. What tells the pump to start?
Primarily three things. Light hitting the cells in the morning, the internal reduction of CO2 because photosynthesis is kicked off, and the plant's own internal circadian rhythm.
As a schedule.
It has a schedule. Conversely, if the plant experiences drought stress, the hormone abscisic acid, or ABA, signals the guard cells to rapidly lose that K plus A, leading to osmotic water loss, loss of turgor, and immediate stomatal closure.
And when plants face extreme environments, they evolve some really dramatic adaptations. We're talking about xerophytes, the dry adapted plants.
Right. They've evolved to minimize that crucial trade-off. Think of an oleander plant. It has a very thick cuticle, which reduces evaporation from the surface, and its stomata are sunk into these small, sheltered pits called crypts.
To protect them from the wind.
Exactly. And these crypts are often lined with trichomes, with those little hairs. This whole architecture creates a small layer of still, humid air right above the stoma, which significantly reduces the rate of water vapor diffusion and loss in hot, dry wind.
So let's shift gears to sugar transport or translocation, which happens in the phloem. Unlike water, sugars move from a source to a sink.
And that's the key distinction. A sugar source is where sugars are produced, like mature leaves, or where they're mobilized from storage, like a tuber breaking down starch.
Okay.
And a sugar sink is where they're consumed or stored, so growing roots, developing fruits, or dormant buds. And importantly, the direction of flow can reverse seasonally.
And this movement is driven by pressure, which is explained by the pressure flow hypothesis. Can you walk us through the steps?
It's a really elegant system built on gradients. So first, at the source, the dominant sugar, sucrose, is actively loaded into the sieve tube elements and their companion cells.
This takes energy.
It takes energy. This process, often powered by H plus sucrose co-transporters that use a proton gradient, is called phloem loading. This massive influx of solute drastically reduces the water potential inside the phloem.
Which means water is going to rush in.
Exactly. Water flows osmotically from the nearby xylem right into the phloem, and this creates high positive pressure, the pressure potential.
Okay.
Then, at the sink, that sucrose is unloaded, either actively or passively, for consumption or storage. This unloading raises the water potential, causing water to flow back out into the xylem. And this pressure differential, high pressure at the source, low pressure at the sink, is what drives the bulk flow of the phloem sap rapidly toward the sink.
We've established the transport systems, but what exactly are the roots mining from the earth? We need to understand the complex chemical dynamics of the soil.
Soil chemistry is a constant push and pull. The key fact to remember is that soil particles generally carry a negative charge.
Okay, that's important.
It's essential because it means that positively charged mineral cations like potassium, calcium, magnesium, they adhere tightly to these particles. This prevents them from being washed away or leached by rain. The soil acts as a cation reservoir.
So how does the root break that bond between the cation and the soil particle to capture it?
They use a really clever process called cation exchange. The roots actively acidify the soil's solution. They do this by pumping out H plus ions and by releasing CO2 from respiration, which forms carbonic acid.
So they're changing their own environment.
They are. And these excess H plus ions, being positively charged, they displace the mineral cations from the negatively charged soil particles. Once they're displaced, the mineral cations are free in the soil solution to be absorbed by the root.
So what about the anions, the negatively charged ions like nitrate or phosphate?
Well, since they don't bind to the negatively charged soil particles, they are highly susceptible to leaching. This is why nitrogen and phosphorus so often need to be replenished in agricultural soils, and why they're major components of fertilizers.
Now, using hydroponic culture, scientists identified 17 essential elements that plants need to survive. And we divide these into macronutrients and micronutrients.
Right. Macronutrients are required in large amounts. They're the structural elements like carbon, oxygen, and hydrogen, plus nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium. Nitrogen is the absolute workhorse. It's essential for nucleic acids, proteins, chlorophyll.
Pretty much everything.
Pretty much everything. And then micronutrients are required in just trace amounts. They typically function as enzyme cofactors, things like iron or zinc, and they often act as the essential pivot point for metabolic reactions.
And if a plant is lacking a nutrient, it tells us. But the location of the symptom, whether it's on old leaves versus new leaves, is a diagnostic clue, isn't it?
It is, and it all depends on the nutrient's mobility within the plant. If the nutrient is mobile, like nitrogen, phosphorus, or potassium, the plant will salvage it from older, established leaves and transport it to the critical, newer growing parts. So the deficiency shows up in the older leaves first.
And if it's immobile?
If the nutrient is immobile, like iron, the plant can't move it. So the deficiency symptoms will appear first in the younger leaves because they can't get what they need from the older parts of the plant.
So beyond the chemistry, we have to discuss the biology. The plant doesn't exist in isolation. It lives in the rhizosphere, which is teeming with microbial life.
The rhizosphere is that thin layer of soil immediately surrounding the roots, and the roots enrich this layer by secreting sugars and amino acids, basically feeding a massive community of rhizobacteria.
And the bacteria repay the favor.
They do. They repay the favor by promoting plant growth, producing antibiotics to defend against pathogens, and increasing nutrient availability. It's a very active ecosystem.
And the most famous mutualism involves the most critical nutrient of all, nitrogen. Plants can't break the triple bond of atmospheric N2. That job falls to bacteria in a process called nitrogen fixation.
This is an incredibly energy-intensive process. It's carried out by bacteria that reduce N2 to ammonia, NH3. The most efficient natural example of this involves legumes like peas and beans and rhizobium bacteria. The plant forms these specialized structures on its roots called root nodules just to house the bacteria.
It builds them a home. So what happens inside that nodule?
Inside, the bacteria transform into what we call bacteroids, and they start fixing nitrogen into ammonium, NH4 plus ricosite, which the plant can use immediately to synthesize amino acids.
And in return?
In return, the plant supplies the bacteroids with copious amounts of carbohydrates. This is why crop rotation planting a non-legume crop one year and a legume the next is such a fundamental, sustainable agricultural practice. It naturally enriches the soil with nitrogen.
The other essential mutualism is with fungi, mycorrhizae or fungus roots.
This is possibly the most critical relationship in plant evolution. It may have been what enabled plants to colonize land 400 to 500 million years ago. The fungal hyphae network vastly increases the surface area for absorption, particularly for water and phosphorus, which is a notoriously immobile nutrient in the soil.
And we have two key structural types here. The distinction is subtle but important.
Yes. Ectomycorrhizae form a dense sheath, or a mantle, over the outside of the root. Their hyphae extend deep into the root cortex, but they only move along the apoplast between the cell walls. They do not cross the plasma membrane.
And these are common in woody plants.
Very common in woody plants like oaks and pines. But the more common type is arbuscular mycorrhizae.
Okay.
They're found in over 85% of all plant species, including most of our crops. They do not form a mantle. Instead, their hyphae penetrate the cell walls of the root cortex cells.
But not the membrane.
Crucially, they do not penetrate the plasma membrane. Instead, they push the membrane inward, forming these complex, densely branched structures called arbuscules within the cell. And these arbuscules are the vital interface for nutrient and sugar transfer.
So the arbuscule structure massively increases the transfer surface area while ensuring the host plant cell remains totally intact. It's a remarkable biological negotiation.
Yeah.
We've built the plant, we fueled the plant, now we move into the culmination of growth, reproduction. And angiosperms, the flowering plants, are masters of this game. They're defined by what the sources call the 3 Fs, flowers, double fertilization, and fruits.
Right. The flower is the specialized reproductive structure. The organs are arranged in 4 whorls. You have the sterile sepals and petals, the male stamens, which produce the pollen, and the female carpels, which contain the ovules.
For life to continue, we need pollination, the transfer of that pollen from the anther to the stigma.
And about 80% of angiosperms rely on biotic vectors. That means animals, chiefly insects. And bees are the most vital.
That draws them in.
They're attracted by colors like yellow and blue, sweet fragrances, and of course the presence of nectar. But they also see the world differently than we do. They can detect ultraviolet patterns on the petals called nectar guides that basically direct them straight to the sugary reward.
Little landing strips.
Exactly. And this dependency drives coevolution, which is a key concept here. The sources shared a really timely example related to climate change.
Oh, the Rocky Mountain bumblebees.
Yes. Researchers found that as floral resources have changed due to climate pressures, the tongues of 2 species of bumblebees have actually shortened by about a quarter over just 40 years.
That's incredibly fast, evolutionarily speaking.
It is. It's a rapid morphological adaptation that reflects the ongoing evolutionary pressure exerted by the plants they pollinate.
So once the pollen lands, the pollen grain produces a tube containing 2 sperm cells. This leads to the unique defining moment of angiosperms. Double fertilization.
This is a highly efficient evolutionary strategy. The first sperm fertilizes the egg, and that forms the diploid, tekenzygote, which grows into the embryo. The second sperm fuses with the 2 polar nuclei in the central cell, and this forms the triploid, 3N endosperm.
And the insight here is the why. Why go through the trouble of fertilizing twice?
Because the endosperm is the food supply for the embryo. By requiring a second fertilization event to initiate food development, the The plant avoids wasting precious resources on ovules that weren't successfully
This
fertilized.
is an efficiency mechanism.
It's an effective investment strategy. It ensures nutrients are only committed to a guaranteed viable embryo.
So after fertilization, the ovule becomes the seed, and the ovary matures into the fruit.
And the fruit's primary job is twofold: protect the seed and aid in its dispersal. The ovary wall develops into what we call the pericarp. The ripening process of fleshy fruits is incredibly complex, heavily influenced by hormones. It causes color changes, enzyme digestion that leads to softening, and the conversion of starches and acids into sugars, all designed to make that seed dispersal vehicle appealing to animals.
While sexual reproduction ensures variation, plants also utilize asexual or vegetative reproduction to create genetic clones.
Asexual reproduction is often just an extension of their indeterminate growth capacity. You see examples like fragmentation, like the little plantlets that sprout from kalanchoe leaves or the eyes on potatoes.
Right.
The advantages are clear. 100% of the successful genome is passed on, you don't need to rely on pollination, and the resulting progeny is often more robust than a delicate little seedling.
But if they rely on sex for variation, and they can't move to find a mate, how do they ensure they avoid self-fertilization or selfing?
They employ some fascinating tricks. I mean, besides having separate male and female plants or having parts that mature at different times, the most widespread mechanism is self-incompatibility.
Okay, what is that?
This is a plant's ability to recognize and reject its own pollen. It's often based on if the alleles on the pollen grain match those in the stigma tissue. The rejection can be dramatic, sometimes involving the enzymatic destruction of the pollen tube's RNA as it tries to grow.
Yeah. So let's circle back to the topic we started with, plant biotechnology and genetic engineering. We established that humans have always modified plants, but modern methods are so much more precise and can cross boundaries.
Modern GMO techniques allow the transfer of specific, desired genes from any organism, not just close relatives, and the goals are profoundly important for human welfare. One goal is reducing global malnutrition.
And that's where biofortification comes in, with things like golden rice.
Exactly. Golden rice was engineered to express genes that allow it to produce beta-carotene, which is a precursor to vitamin A. This is specifically designed to combat vitamin A deficiency, which causes millions of cases of blindness globally every year.
Another key success involves pest control, specifically with beet maize.
Beet maize contains a gene from the bacterium Bacillus thuringiensis, which produces a protein that is toxic to many insect pests, and is highly targeted because the protein only becomes active in the alkaline guts of insects, making it harmless to vertebrates.
And there's a secondary benefit there that's often overlooked.
There is. Maize kernels contain up to 90% less fumonisin, which is a potent fungal toxin and a known carcinogen, because the reduced insect damage prevents the fungus from infecting the kernels in the first place.
And looking toward our future energy needs, biotech is playing a role there too.
Oh, yeah. To combat fossil fuel dependence, scientists are engineering fast-growing biomass crops like switchgrass and poplar to make them more efficient at converting their structural components, cellulose and hemicellulose, into fermentable sugars for biofuels. They are literally redesigning plants to be better fuel sources.
Of course, genetic engineering comes with controversies. We should acknowledge the main environmental fears, starting with the concern about non-target organisms.
The famous example there involved the monarch butterfly caterpillars. An initial, heavily publicized lab study suggested that bait pollen harmed them.
I remember that.
Right. However, that study was later shown to be flawed. It used artificially high pollen concentrations, and it was actually other floral parts, not the pollen, that contained the toxin. Subsequent field research has generally confirmed that conventional chemical spraying is far more harmful to monarch populations than the targeted bee white crops are.
So the most serious ecological fear that remains is transgene escape. The fear that herbicide resistance genes could transfer via pollen to wild relatives, creating herbicide-resistant superweeds.
And this is a legitimate risk that demands mitigation. Scientists are developing three main strategies to prevent this escape. First is engineering male sterility so the plant can't produce functional pollen.
Okay.
Second, engineering the desired gene into chloroplast DNA, which is inherited only through the egg, not the pollen. And third, engineering flowers that are self-pollinating and never open, preventing pollen release to the outside environment. The science is really focused on balancing the extraordinary benefits of this technology against these real ecological risks.
We saved this section for last because it absolutely demolishes the myth that plants are passive organisms. They're constantly receiving, interpreting, and responding to their world. And the mechanism for this is the signal transduction pathway.
Everything a plant does in response to its environment, whether it's bending toward light or triggering a defense response, begins with this pathway. Let's use the classic example of de-etiolation or greening up.
That's when a dark-grown seedling, which is pale and spindly, is suddenly exposed to light.
Exactly. So step one is reception. What catches the light signal?
The photoreceptor.
The photoreceptor phytochrome, which is located in the cytoplasm. Phytochrome is a molecular switch that changes its conformation when it absorbs red light, and that initiates the whole signaling process.
Then step 2 is transduction. How is that tiny light signal turned into a massive change in the plant's growth?
This is where second messengers come in. The signal is amplified using internal cellular messengers, specifically increasing cytosolic calcium ions, Ca2 plus per Menke, and stimulating the production of cyclic GMP, or cGMP. These messengers then activate cascades of protein kinases, which phosphorylate and activate target proteins, leading to profound changes in gene expression.
So the plant has moved from a signal to amplification to the final response.
And the response is immediate. Stem elongation slows way down, the leaves begin to expand and unfurl, and the genes for chlorophyll production and photosynthesis enzymes are activated. Crucially, the levels of growth-promoting hormones like auxin and brassinosteroids are altered, slowing down that vertical growth now that the light has been found.
This moves us perfectly into plant hormones, the chemical messengers that coordinate growth and function throughout the entire plant, often working at unbelievably low concentrations.
And their effects are complex. A response often depends not just on the absolute concentration of one hormone, but the ratio of several hormones interacting at the same time.
Let's start with auxin, or IAA, the classic growth hormone. It's synthesized primarily in the shoot apical meristems.
And auxin's transport is critically important. It's unidirectional or polar transport, moving from the tip down to the base, mediated by special basal auxin transporter proteins. It's what controls phototropism bending toward light by stimulating cell elongation on the shaded side of the stem.
And this elongation mechanism is what we call the acid growth hypothesis. Wait, so are the cells essentially dissolving their own walls to get longer?
That's a really good analogy for the physical process. Auxin activates plasma membrane proton pumps, which pump H+ ions into the cell wall, lowering its pH.
Making it acidic.
This acidic environment activates proteins called expansins. The expansins break the crosslinks within the cell wall cellulose, which loosens the fabric. Once it's loosened, the high turgor pressure inside the cell pushes the cell out, causing irreversible elongation.
So auxin gives the instructions, the protons create the environment, and turgor provides the force.
That's a perfect summary.
We mentioned auxin's role in apical dominance earlier.
Yes, the auxin coming from the apical bud inhibits the growth of the axillary buds. But it's not just auxin working alone. Auxin actually works by inducing the production of another class of inhibitory hormones called strigolactones, which are what directly suppress the bud development.
And the antagonist to that repression, what encourages branching?
That would be cytokinins. They are synthesized mainly in the roots and move up through the xylem. They act as counterbalances to auxin and strigolactones, stimulating cell division and differentiation. We see this in tissue culture all the time. A high cytokinin to auxin ratio leads to shoot formation. The inverse ratio leads to root formation.
And they have an anti-aging effect too, right?
A powerful anti-senescence effect, yes. They mobilize nutrients and help keep tissues young.
Next up, gibberellins, or GA, known for promoting dramatic stem elongation and seed germination.
GA causes what we call bolting, that rapid growth of internodes that often precedes flowering. Commercially, this is used to enhance products like seedless grapes, where GA application increases both the fruit size and the spacing between the grapes in the cluster.
And in seeds.
In seeds, after water is imbibed, the embryo releases GA, which signals the aleurone layer to produce digestive enzymes like alpha amylase to break down the stored starch, which fuels germination.
Then we have the two major stress hormones. First, abscisic acid, or ABA, the brake pedal.
ABA is the primary slow-growth hormone. It promotes seed dormancy, ensuring the seed only germinates when conditions are truly favorable by inhibiting metabolic processes. Its critical short-term role, however, is in drought tolerance. When the plant senses water stress, ABA levels rise and rapidly signal the guard cells to close the stomata, minimizing water loss.
At the cost of temporarily slowing photosynthesis.
Exactly. And the final classic hormone, the gaseous messenger, ethylene.
Right.
Ethylene is produced in response to stresses like injury, drought, and especially flooding. If a seedling hits an obstacle underground, ethylene mediates the triple response: a pause in vertical elongation, a thickening of the stem, and horizontal growth, which allows it to navigate around the obstacle.
And it's also the hormone of aging and leaf drop.
It governs senescence and leaf abscission. When the ethylene to auxin ratio gets high in the fall, it triggers the formation of the abscission layer, sealing the leaf off and salvaging key nutrients before it falls.
Moving beyond chemical messengers, plants also have these highly refined responses to physical stimuli. How do they detect gravity, which guides their growth?
This is gravitropism: roots growing down, which is positive, and shoots growing up, which is negative. The detection mechanism involves statoliths. These are dense, starch-filled plastids located within specialized cells of the root cap.
Like little weights.
Exactly. And as these statoliths settle to the lowest point of the cell, they trigger the lateral transport of auxin to the lower side of the root.
And because high auxin inhibits root growth, the lower side grows slower and the root bends down.
Precisely. The upper side, with less auxin, grows faster, pushing the entire root tip downwards. It's a beautifully simple feedback loop.
They're also sensitive to touch. Thigmomorphogenesis is the structural change from chronic mechanical stress.
You see this in nature all the time. Trees growing in consistently windy areas are characteristically shorter and stockier, with thicker trunks, compared to sheltered trees of the same species. They're responding to the friction and stress by prioritizing girth over height.
And then there's thigmotropism.
Which is the directional growth response, like a tendril coiling around a trellis, responding dynamically to contact.
Some plants also exhibit surprisingly fast movements, like the Mimosa putica folding up when you touch it.
That rapid response, which happens in a second, is mediated by action potentials. They're electrical signals, though much, much slower than in animals. The touch triggers an action potential which travels to these specialized motor cells called pulvini.
Okay.
The pulvini rapidly lose turgor pressure by pumping ions out, which causes rapid osmotic water loss, and the resulting change in shape causes the leaves to instantly fold up.
So let's end with the plant's formidable defense systems against pathogens and herbivores. They are not helpless targets.
Not at all. Plant immunity operates in two layered systems. The first line of defense is PMP-triggered immunity. PMPs are generic pathogen-associated molecular patterns, like a common molecule found on the surface of most bacteria. Plant receptors recognize these generic threats, and that triggers the production of antimicrobial chemicals called phytoalexins and general cell wall reinforcement.
But pathogens fight back using their own specific weapons.
They do. They deliver proteins called effectors that are designed to cripple the plant's PMP defenses. And this forced the evolution of the second line of defense, effector-triggered immunity, or ETI. Plants have hundreds of R proteins, or resistance proteins, each one tailored to recognize a specific pathogen effector.
And the recognition of a specific effector triggers a massive, localized counterattack.
The hypersensitive response. This is rapid, localized, programmed cell death right around the infection site, which forms visible lesions. By killing a small cluster of its own cells, the plant starves the pathogen and prevents its spread throughout the vascular system. It's a local sacrifice.
A desperate, effective measure. And this localized response also alerts the rest of the plant, doesn't it?
It does. The localized hypersensitive response generates chemical signals, including the hormone salicylic acid, which is transported throughout the plant body. This induces systemic acquired resistance, or SAR. It's a generalized, nonspecific defense that protects the entire plant against a wide variety of pathogens for several days or weeks, giving it time to recover.
And finally, against the macroscopic attackers herbivores, the defense is multi-layered, spanning physical structures and complex chemical warfare.
Chemical defenses include molecular bombs like terpenoids, some of which mimic insect hormones and disrupt their development. You have phenolics, like tannins, which taste bitter and interfere with digestion, and of course alkaloids, like the powerful psychoactive substances found in the opium poppy.
What about physical and cellular defenses?
We see tough, supportive sclerenchyma fibers making leaves really difficult to chew. At the cellular level, some plants house specialized cells called idioblasts that contain these needle-shaped calcium oxalate crystals called rhyphides.
Oh, this sounds nasty.
It is. When the plant tissue is chewed, these tiny, sharp needles act as carriers, piercing soft tissue and delivering chemical irritants right into the herbivore's mouth.
And the highest level of defense actually involves recruiting help from the surrounding ecological community.
Yes, the plant acts as a communication hub. When it's attacked by a caterpillar, for instance, the plant releases specific volatile chemicals into the air. These chemicals are essentially distress calls, signaling specific predatory parasitoid wasps to fly in, locate the caterpillar, and lay their eggs inside it, killing the pest.
So it's actively calling for backup.
A remarkable and active strategy of community defense. Hashtag tag tag outro.
We have journeyed through the incredible structure of meristems, the physics of water transport, the chemical warfare of defense, and the hormonal choreography of growth. The core takeaway for me is clear. Plants are profoundly active organisms. They're constantly signaling, sensing, and dynamically optimizing their structure and function.
And everything we've discussed, from the cohesion-tension mechanism to the pressure flow hypothesis, it all relies on carefully managed gradients, whether they're gradients of pressure, water potential, or hormone concentration. The elegant simplicity is that structure is always perfectly tuned to function.
As we close out this deep dive, let's leave you with one final thought that forces you to connect the external world, the plant's internal timing, and its chemical responses.
Right. We detailed how plants use phytochrome to detect light quality and use that information integrated with their circadian clocks to time critical seasonal events like flowering, a process we call photoperiodism.
Okay.
Now, imagine a catastrophic global event, let's say a major volcanic eruption that darkens the entire globe, removing significant external light signals for 3 consecutive months.
So how would this sudden, prolonged absence of light signals interact with the plant's ingrained circadian clock and that phytochrome switch? Would the internal clock, which is expecting light to initiate photosynthesis and regulate hormone production, just become completely decoupled and fail?
Or would the sudden darkness immediately trigger a high-stress response, forcing massive ABA production and perhaps a premature, fatal dormancy, regardless of what the internal clock's dictates are? And you have to consider the long-term evolutionary consequences. Would annual plants, which have to complete their reproduction within a single season, be completely wiped out, while perennials, with their deep root systems and years of energy reserves, might have a better chance of survival?
Something to mull over as you observe the greenery around you. Thank you for joining us for this deep dive into plant form and function.